Staining of blood smear. Usually two types stains used in blood smear staining, Giemsa stain and Leishman stain. Staining of blood smear, Leishman stain. This stain has a pure keeping quality in hot climate, so it is better to prepare a fresh stain from the powder every month. Composition: Leishman powder, isonimethyl blue powder 0.5 gram, acetone free methyl alcohol 100 ml. Staining of blood smear. Leishman stain. Prepare blood smear, air dry it. Pure Leishman stain dropwise, counting the drops on the blood smear and allowed to act for 1-2 minutes. Add double quantity of distilled water dropwise over the blood smear. Double the number of drops. Staining of blood smear. Leishman stain mixed by the rocking the slide gently. 15 minutes are allowed for staining. Slide is washed by the stream of distilled water horizontal position until the smear looks pink. Thereafter the slide kept in the vertical position so that drains the dries. Staining of blood smear. Leishman stain staining procedure. Dry in air and examine under the oil immersion lens of microscope. Fixation is not required in Leishman staining procedure because methyl alcohol is already mixed in Leishman stain. Staining of blood smear. Leishman stain microscopic examination. Platelet, erythrocytes and leukocytes. Staining of blood smears. Giemsa stain. Staining procedure. Prepare blood smear and air dry it. Fixation in the methyl alcohol for 2 minutes. Take out, dry it, pure. The diluted Giemsa stain 1 in 10 dilution over the fixed smear. A part of Giemsa stain plus no part of distilled water, buffer water. 1 part Giemsa stain plus 9 part distilled water or buffer water. Staining of blood smear. Giemsa stain, it is kept for 45 minutes for allowing stain. Wash the smear under distilled water, tap water till the smear is just pink. Stained blood smear are dried in the air and examined under the microscope under oil immersion. Staining of blood smear. Giemsa stain microscopic examination. MST got of L. Donovani, bone marrow Giemsa stain. Application of Giemsa stain. Staining of blood smear. To avoid this, the slides should be placed vertically into the Kipling jar, having the required stain preferably stain on the horizontal rack. Diluted wash with the buffer in the same position. If the stain is poured off, the scum in the label to stick to the slide from artifacts. Blood smear preparation not technique. It is used for the diagnosis of microfilaria, Dirophilaria, emetis, diptyloneuma species through the lysis of the red blood cells. Staining blood smears, not technique. Suspected hepatitis blood 2% formalin methyl blue glass slide cover slip centrifuge tube pipette. Staining of blood smear, not technique. Collect the blood from the suspected dog in the hepatitis vial. Mix 1 ml hepatitis blood, 3 ml 2% formalin. Centrifuge the formalin, mix the 1500 rpm for 5 to 8 minutes. Discard the supernatant and then desediment. Not techniques. 1 drop of mixture, place glass slide in the help the pipette and apply cover slip over it. Examine microfilaria under the 40x 100x. Staining of blood smear microhematocrit centrifuge technique. It is used for the detection of microfilaria of filarial worms trypanosoma species parasite. Staining of blood smear microhematocrit centrifuge technique. Suspected blood sample microhematocrit tube sealing materials or wax microscopes. Blood smears preparation microhematocrit centrifuge techniques. After the filling the microhematocrit tube with the blood, centrifuge it for 1 minute. When suspected blood contained macrohematocrit tube is the centrifuge. There three layers, example plasma, WBC, Buffy coat layer and RBC layer. Blood smear preparation, microhematocrit centrifuge technique. Under the dissecting microscope, the tube is the place, then the examine and the interface interface of the plasma and fluffy coat motility in the plasma microfilariae will be observed in active swimming condition. Blood smear preparation. At the Buffy coat interface, break the tube and tape is the Buffy coat and small amount of plasma on slide. After the placing a cover glass live macrofilariae trypanosoma species can be examined. Allow it to dry. Fix for the minute with methyl alcohol stain with Giemsa stain 30 minutes for identification. Blood smear preparation. Microhematology centrifuge technique. Trypanosoma species in microfilariae may be observed in the Buffy coat smear after microscopic examination. Staining of blood smear, lymph gland biopsy. Lymph node smear is the sometimes used in the preference to blood smear. For example, during investigation, sclerosis and even trypanosomasis, common lymph node used for the biopsies prescapular. Staining of blood smear, lymph gland biopsy, glass slide. Materials required: glass slide, saving blade, methylated spirit, and needle. 1618 gauze for cattle. Staining of blood smear, lymph gland biopsy. Staining of blood smear, lymph gland biopsy. Cyzone stained, coast blue bodies, Claria species found inside the cytoplasm, lymphocytes, or